This tutorial is designed as a guide for the DNA collection of casework known samples. A known sample is DNA that we know comes from a particular person. We use these to compare to any unknown profiles we develop from items of evidence. A known DNA sample can come in a few different forms, with the most common being known buccal swabs, which is what we will be focusing on in this video. When we say buccal, we are referring to the cells of the inside of the cheek. So a known buccal swab is simply the swab of the inside of an individual's cheek. If you're looking for the video on how to collect samples for the database section, please visit the Profiling Convicted Offenders page on this website. Do not use database FTA cards to collect known DNA samples for casework. If available and applicable, known DNA samples should be submitted for every case. The most helpful knowns to have when submitting evidence would be from the victim, the suspect, the consensual sex partner of the victim and/or suspect for sexual abuse cases, other individuals whose DNA may be on the item of evidence, and both parents of an offspring in a paternity case. You will need to gather a few items before collecting known DNA samples for casework. Clean gloves, 2 packages of single-use cotton tip swabs, preferably DNA-free swabs. If DNA-free swabs are not available, sterile swabs are also okay. Swab box. External packaging materials, markers, and evidence tape. First, start by putting on the pair of clean gloves. This helps protect you and prevent any contamination. Use your marker to note on the swab box whose known buccal sample you are collecting and which case they are associated with. The DCI Crime Lab uses the name of the individual, the case and item numbers, the date collected, and the initials of the collector. If your agency has other marking requirements, then use those. Next, open up the swab box and close one end. Carefully open the swab packages and take out the 2 swabs. Instruct the individual to open their mouth and place your hand on the outside of their cheek. Using a generous amount of pressure, swab the inside of one cheek with 2 swabs. Make sure to twist the swabs around to ensure all parts of both swabs get an equal distribution of buccal cells. With the same 2 swabs, move over to the other cheek and repeat this process. Once finished, place both swabs in the prepared swab box in the same direction. Close the remaining end. Apply evidence tape to both ends of the swab box. At the Crime Lab, we use date and initials when sealing swab boxes, but use your agency's requirements. You are now ready to put the swab box into the external packaging material and submit it to the lab. If you have any questions, please contact the DNA section of the Iowa DCI Crime Lab at 515-725-1500. Special thank you to the DMAC video production class for filming and putting together this video.